Okay, so this video, Dr. Roos, this is a video I'm doing about DNA replication, transcription, and translation. I'm going to put all three of them together just to give you a quick overview of what they all entail. So the first one, DNA replication, is when a cell or an organism, it could be a bacteria that divides, it could be a cell that divides, right? And the genome of that organism, and I have a video on the genome of that organism needs to be duplicated, replicated, sometimes it's synthesized is used, the word. But you want to go from one copy to 2 copies when you're done. So that if a cell goes from being 1 cell to 2 cells, right, then each cell has a copy of all your genetic information, your genome. Now, in most textbooks, you have what's called initiation, elongation, and termination of all 3 of these. And initiation means it begins or starts here. Elongation means you're making DNA, you're making RNA, or you're making protein, and then it stops when it's finished. Okay, so that's termination. So with DNA replication, we need to start at what we refer to as an origin of replication is usually the start point that they refer to it as. And then you need to use an enzyme. And fortunately, when you're making DNA, the enzyme you use is called DNA Polymerase. Okay, that's the enzyme that produces DNA. So another way of putting it is DNA polymerase uses your original DNA template as a template to make another copy of the DNA. Right, it's a long strand. And then to make the DNA, you have to, the substrate for this enzyme is called little d for deoxyribonucleic acid, which is what DNA stands for, NTPs. And NTP is a nucleotide triphosphate, right? And there's 4 of them, right? There's 4 letters in DNA. So there's a little d ATP, there's a little d CTP, there's a little d GTP, little d TTP. So these 4 nucleotides are polymerized by DNA polymerase to make another copy of DNA. It initiates, it begins at origin of replication, right? That's the point in the DNA that you start at. Okay, and we're using these nucleotides to produce DNA. So with transcription, we're going to basically, this is a different ball game, right? DNA replication, make a copy of your genome. Transcription and translation refer to the process of gene expression, right? And I have a video about what a gene is, right? It's a piece of DNA that has the genetic information to make a protein. We call that gene expression when we use transcription and translation, right, to turn the genetic information into a protein. So for genes, DNA replication, transcription begins at the promoter. Okay? The enzyme that's going to make the RNA is called RNA polymerase. Okay? DNA polymerase makes DNA, RNA polymerase makes RNA. There's actually 3 or maybe even more polymerases. I think there's like 3 RNA polymerases too. Right? And I need to make a video probably just talking about those enzymes. But right, this makes RNA, these make DNA. So to make the RNA, converting the DNA to RNA, we need RNA nucleotides, or NTPs, without the little d. And the four NTPs are ATP, CTP, GTP, not TTP, but UTP. Right? A, C, G, and U are found in RNA, right? You say the U replaces the T that's found in DNA. These nucleotides can't be used to make RNA. These nucleotides can't be used to make DNA, right? The little d means there's a deoxyribonuclease, or deoxyribose is the sugar in these nucleotides. Ribose is the sugar in those nucleotides. That's the difference. ATP is what you make when you have cell respiration in the mitochondria. It's the big, say energy molecule, like a little battery. But all these nucleotides can be used for energy just like ATP. Okay, so once you make the RNA, right, you've expressed, the promoter is, say, like the on-off switch. So if it's turned on, RNA is made. Okay. Now you take the RNA to the ribosome, right? That's where protein synthesis occurs. Right? The ribosome makes the protein. Okay. So you make what's called the translation initiation complex. So it's referred to as a complex. And we're going to, convert at the ribosome, we're going to convert the RNA into a protein. Right, so in so doing that, we are using codons that are found in the messenger RNA. We're going to, at the ribosome, the anticodon is going to bind to the codon. The anticodon is what the transfer RNA has, right? So, say if this is a codon, right, A, G, C, then the anticodon would be U, C, G. That's found in the tRNA, right? All tRNA contains, right, an amino acid attached to it. So, the transfer RNA, tRNA, binds to the messenger RNA by binding the codon in the messenger RNA with the anticodon in the tRNA. That allows the tRNA to bring in the correct amino acid. So this codon tells the ribosome that so-and-so amino acid belongs in the protein. Okay. So the genetic information that's contained in these nucleotides, right, are found in these codons, and they are, like, say, the recipe that allows the ribosome, right, to make the protein. Right, so a protein is composed of amino acids. There are 20 different amino acids, and you have to put them in the correct order, right? The correct sequence is another way of putting it, right? So the sequence of these nucleotides that are originally in the DNA become a sequence of nucleotides in RNA. You translate the nucleotide sequence, A, G, C, U's, right? That's translated into a sequence of amino acids to assemble the protein, right? You polymerize the amino acids, and they have to be in the correct order. So what I wanted to do in this video is basically make it clear what these three concepts do, right? If you're replicating DNA, you're using DNA to make DNA. For transcription, you're converting DNA into RNA, and in translation, messenger RNA is converted into protein. Gene expression means, right, you're converting the gene, the genetic information of the gene, into a protein. Okay, this is, say, the hard drive information is the nucleotide sequence, and that's used to assemble the amino acids together, and those amino acids generate a protein that can get jobs done in cells or biological systems, right? They become, whatever components we need in our body to have our muscles move or contract, to visually see things. Proteins are what, for the most part, get the job done. There are other things involved, but right, they're the backbone.